अंकल अंकल अंकल, आपकी राइटिंग बहुत बुरी है।
Production.
चला दो माइक।
What's the presence? What would the presence be? Well, if, for example, I have too much of something, right? Let's say the polymerase keeps going around and around and around and starts making, let's say, a whole bunch of testosterone in your body. If you get a whole bunch of testosterone in your body, it's going to increase in concentration. And the increase in concentration increases the likelihood that it binds to the repressor molecule, thereby binding to the operator and thereby inhibiting. The presence represses or inhibits the production. So that's where the repressor molecule is not always attached. But how about if we have the repressor molecule where it always is attached? Here's our polymerase. Again, this is the promoter, the operator, the gene. Notice how the polymerase cannot pass the operator region because of the presence of the inhibitor molecule. In this example, the presence induces production. Can you understand? You see what I'm writing? You guys good? Someone say yes. Okay, thank you. So the presence induces production. What does that mean? That means that if the person decided to have milk, here it is, here's the lactose. The lactose binds, makes this fall off, thereby allowing the polymerase to pass. And when the polymerase passes, it gets to the genes that code for making the enzyme lactase. Then the enzyme lactase will be assembled, it will be made. And then the enzyme itself will start to digest the lactose. And when it eventually digests this one, this guy will go back on, repressing or stopping the production. So the presence induces the production. What is the presence? It's usually something that you're eating. This is an example of where you are making enzymes. And the other example is when you're always making something in your body, you always need it, but then your body's making it. It's already now getting too much, then your body wants to stop it. And how does it stop it? It will attach to the repressor molecule and inhibit the production. So that's the crux of the lesson. This happens all over the body. And remember, this is the Ocron model. So it happens in I'm going to say make a little bit more sense. I mean, I hope that my lesson will make it more clear. So what I've done is I've categorized the mutations as either being a point mutation or a chromosomal mutation. A point mutation is a mutation that occurred on the strand of chromosome, let's say in a cell, in a somatic cell. So that mutation could result in a mutation in your toe, or a mutation in your finger, right? It's not the whole organism that is exhibiting the mutation, whereas the chromosomal mutation is the mutation that's occurring when the gametes have come together to produce the zygote. And as such, the whole individual is exhibiting that mutation. But does that, first I want to make sure that yeah, exactly, like cancer. See if I can jump back to the screen. I would not know. Exactly like cancer. So you could get a growth in one part of your body, but it's not the growth is all over, unless metastasis occurs, and then that's the spreading of that cancer. So far, so good? Good. So now, I'm going to say, let's take a look at just point mutations for now. I also want to say, this note is an awesome note or template for what you can expect on your upcoming unit test. Because what I'm going to do is, I'm going to give you the original strand, I'm going to give you the mutated strand, and I'm going to ask you, okay, what's the complementary strand? So like I'll put, let's say this is the 3 to 5. Here I didn't specify 3 to 5 or 5 to 3. But I'll say, okay, so what's the complementary strand? And you'll give me the complementary strand. And then you'll give me complementary strand. And then I'll say, okay, well, from reading the 3 to 5 direction and assembling the 5 to 3 direction, which you've already made, or the complementary strand, what is the messenger RNA? So you would need to know how to make me the messenger RNA. Remember, the messenger RNA strand is always going to be, if this is the 3, which it is, this is the 3, the opposite of that is going to be the 5 strand, and the 5 strand of the messenger RNA is the exact same as the 5 strand of the DNA, with the only addition or difference rather of the uracil. So notice that this is T, this is A, right? That means that this is the opposite. And if that means it's the opposite, that means this has got to be 3 to 5 direction. Because if it was 5 to 3, this strand would be exactly like that, just the difference is uracil. Does that make sense? Do you see what I'm saying there? Okay, good. Mostly. Yes. Okay, so I'm going to allow me then just to kind of repeat it. How do I see I didn't put down the 3 to 5? How do you know it's 3 to 5? Well, if you look at the messenger RNA, the messenger RNA is going to look exactly like the DNA's 5 to 3 strand. Right on this thing.
रिकॉर्डिंग होती है मैं के बाद में देख हम हमारे रिकॉर्ड टीम्स पे तो रिकॉर्ड करने की जरूरत ही नहीं होती
A, T, C, G.
G2
If this is the DNA, I'm saying it's 3 to 5.
लाइव है या रिकॉर्डेड
Then the complement, the 5 to 3, has to be
128
T, A, G, C. And then if I ask you, well, what is the mRNA? mRNA is going to be the polymerase reading the 3 to 5 direction and assembling the 5 to 3, which is the same thing as this. The only difference here is it's going to be uracil, adenine, guanine, cytosine. So the mRNA is exactly like the 5 strand of the DNA, but the only difference is it gets rid of the thymine and replaces it with uracil. Here we go. Now, does that make more sense? Beautiful. I need to stop sharing for a reason. test what type of mutation. Be specific. Don't say it's just a point mutation, because there's a lot of different types of point mutations, right? So really what you want to do is you want to say to me, is it a silent, a missense, a nonsense, an insertion, or a deletion mutation? Okay, so that's important. Oh, 1 second. Hold on. Cannot be 5 to 3. Why? Because the messenger RNA is not exactly like that guy there. It's the complement, which means this is 3 to 5. The 5 to 3 is not shown. That's the 5 to 3, just with the replacement of the uracil. Yes? You sure, you sure? Okay, good. So, that's better. Oh, no problem. Okay, so, so now here, there is one base substitution. This is the one base substitution, as you can see. So what effect does that have? So I'll give you this. You may need to give me the complement. You'll definitely need to give me the messenger RNA, and then you'll definitely need to give me the amino acid sequence, and you'll definitely need to tell me what kind of a mutation it is. So how do you do that? Well, once you finish getting your messenger RNA, and you put them in codons, then you'll go to your chart, which I will absolutely give you on the text. So codon table, oh, there we go. And I'll say to you, okay, what are the amino acid sequences? And so you will go to, I'm going to just focus on this one because this is where the mutation occurred, right? So AAC, A, and then there's the second A. So AAC is ASN. Now, if you go back and you take a look at AUU, which is the mutated one, AAU, it's still the same amino acid. That means that this is a silent mutation. Yes, the mutation occurred, but look, the sequence of amino acids is exactly the same. Meaning, there is no change in structure, and thus there's no change in function. So that's what a silent mutation is. A missense mutation is where you may have multiple mutations. And see this one? It still did not result in a change in the amino acid. Second mutation, right? Codon 1, codon 2, codon 1, codon 2. When we go to the messenger RNA, codon 1, codon 2 means that it's this amino acid, right? And this guy is that amino acid, the one above. We just added it. We just did the same. See? No amino acid sequence change. That's okay. But if there is one mutation that results in a different codon, which results now in a different amino there we go, in a different amino acid, that means that the sequence has changed. If the sequence has changed the structure, the function, this is known as a missense mutation. The last example is known as a nonsense mutation. A nonsense mutation means that the stop codon appears prematurely. So there is a single base change that just so happened to result in an early stop codon, which means everything after that is not even included. This is why a nonsense mutation usually results in, most likely, a non-functional protein. This one could be a semi-functional, depending on how bad the structure has changed, but this is even worse. Follow me? Oh boy. to give yourself a chance to digest this, meaning like after a lesson, right? So what happens to the rest? There is no rest. Good question, Zainab. Because the early stop codon appears, the polymerase just stops there and doesn't even continue going on. It thinks that there's nothing else to read. That's why there's a much more significant change in the structure. And so the frame shift mutation is actually just kind of like the name. It's a deletion. So here is the original, and the original C is gone. So notice TAC, TAC, TCG, TCG. Notice that there's no C here. That means that we've reclumped, we've regrouped them. That means that because that C is gone, these guys now got regrouped, and that's why you see that there. And then these guys got regrouped into a codon that you see there. And that's why we ended up with not even a codon at the end. So literally, it's the delete. And as you can see, it has this effect in the change of amino acids. Yes? Yes. The opposite of a deletion is just the insertion, and it's also going to have that domino effect. So in the insertion, we are adding a G. Notice that there is no G here. So when we added a codon, it now caused the clumping of everything as well. So we added a G here. Instead of the T, C, G being together, now we've split it up. So we've added the G. So there's a G here, so that becomes a GTC. GTC, which means now this G has to be reclumped like a domino effect. These three get reclumped, and that's where we see that there. And then the GT is that right there. Again, ending up with no codon at the end as well, hence the question mark. And that's the insertion. The chromosomal mutations, like I said, is happening when the zygote is formed. Translocation is when here's, let's say, mom's and here is dad's. So this is mom's chromosome number 1 and this is dad's chromosome number 1. Mom's and dad's chromosome number 1 are coding for the same thing, but they're not the same message, meaning this is coding for eyes and this is coding for hair. I don't know, right? And mind you, it's not going to be just one characteristic on the entire chromosome, but just for simplicity purposes. So what happens is the information gets flipped on the very same chromosome. So chromosome 1, hair, let's say hair and hair. So they get flipped. Instead of blonde hair, you get brown hair. The information is flipped. It's on the same chromosome. Mom chromosome 1, dad chromosome number 1. Transposable elements is we have 2 different chromosomes. So here is mom and dad's chromosome 1, and here is mom and dad's chromosome 2. Well, chromosome 1, both for mom and both for dad, has to do with eye color, and then this has to do with how brittle your toes are. Toenails, let's say. So the information from eyes goes onto your toes. So it's a larger impact. 2 different chromosomes are involved in here. Here it's the same chromosome. And last but not least is the inversion it's the complementary chromosome. It's the same chromosome, but we've inverted. So look at that. That's D, that's E. Here it's E and D. So it could disrupt transcription because it's flipping the information. And what could cause these mutations? What could cause the inversion, transposable elements, or translocation, or what could even cause the inversion, the deletion, right? The nonsense, missense, or silent mutation. What could cause that? Which could just be spontaneous, meaning that it's just polymerase 1 and 3 they missed it when they were proofreading it. It also could be, let's say, the hormones that are in our food, the pesticides, the herbicides in our water and in our food. Radiation, smoking, drinking, all of that stuff. And then the final thought I threw, like, what do you think would happen if there was no start codon? If there was no start codon on the messenger RNA, what do you think would be a consequence? What do you think would be the outcome? No protein, exactly. So no synthesis would be occurring. It wouldn't know when to start and where to start. So then the next lesson is, then I've been kind of talking about this a little bit throughout anyhow, and it's the difference between prokaryotes and eukaryotes. And I've already mentioned to you that, like, our prokaryotes have those circular plasmids. I think I drew that at the top earlier on. I think I erased it. Oops. Oh, there it is, right? Just finish it. There you go. Okay, so there's your prokaryote. And the cool thing about prokaryotes is that the polymerase can be reading and producing the mRNA at the very same time, and that's all in the same area at the same time, and that's what I'm saying up there. And with us, we have introns, we have non-coding regions, which means I've even in the past, let me ask you, if bacteria do not have introns, but humans have introns, how could this impact bacteria? How could having no introns impact bacteria? And just hear Davion saying, you're making my head hurt. Thanks for the laughs. So let me rephrase it. Like, if you're looking at a bacteria and think about now, when that bacteria is replicating that genetic information, if it has no introns, what do you think would be the impact? Come on, guys, get it wrong now. Zainab, interesting. In fact, it's the opposite, buddy. If they have no introns and there is a mutation, then it's going to happen 100% on the exons, which is the coding region, which means that there's a greater likelihood of mutations occurring in the prokaryotes. Does it make sense now? Anybody else? Thank you, Zainab. Amen, Aaliyah, Antonio, the rest of you as I go down the list here. Bernice, Daniel, Lana. Do you see what I'm talking about? Say no if you don't, and then I know you don't, but all I see is your name on the screen. Okay, thank you. So what I'm saying is this. If this plasmid, here's my bacteria, right? If this plasmid has no introns, that means it has no areas of non-coding regions, which means every little region, every little square inch of this plasmid genetic information codes for something, and therefore codes for something significant, right? And if the polymerase that I just drew here is going along and makes a mistake, then it's going to code for something. And if the polymerase that I just drew here is going along and makes a mistake, then it's going to code for something. And if the polymerase that I just drew here is going along and makes a mistake, then it's going to code for something. And if the polymerase that I just drew here is going along and makes a mistake, then it's going to code for something. And if the polymerase that I just drew here is going along and makes a mistake, then it's going to code for something. And if the polymerase that I just drew here is going along and makes a mistake, then it's going to code for something. And if the polymerase that I just drew here is going along and makes a mistake, then it's going to code for something. And if the polymerase that I just drew here is going along and makes a mistake, then it's going to code for something. And if the polymerase that I just drew here is going along and makes a mistake, then it's going to code for something. And if the polymerase that I just drew here is going along and makes a mistake, then it's going to code for something. And if the polymerase that I just drew here is going along and makes a mistake, then it's going to code for that mistake will definitely occur on an exon because there's no introns, which means it definitely will occur on a coding region, which means there will definitely be a mutation. Whereas if it was in a human, in a eukaryotic cell, here's my nucleus, here's my genetic information, and the polymerase made a mistake when it photocopied the genetic information, it could have done so on an intron, which means that it would not have even had an impact because it was a non-coding region. As that now is, because there's no exons and introns in prokaryotes, there's no spliceosomes because there's nothing to splice. Some other little differences is there is no AUG, there is no start. There is a start, but it's just known as a whole bunch of 5 carbon caps known as the Shine-Delangro sequence. The other thing is the ribosomes are much more simplistic in prokaryotes, and although there is methionine, which is pretty cool, and you're like, okay, whatever, what does that matter? Well, think about it this way. We are big walking protein. All of our proteins start with methionine, and so with prokaryotes. There is the evolutionary link and significance there, okay? They just have an alcohol group attached to it. And like I said, we don't have operons, so the model that we've learned as to how we can affect turning on and off gene production or translation is different in humans. And we talked about the circular chromosome. We already named it as a plasmid. And the endosymbiosis. And that's where I even said to you guys, okay, well, take a look. There must have been an evolutionary link. And the last little caveat today is the evolutionary link as well is we see some single-cell living organisms that even with the mitochondria. Mitochondria gave up its life to be an organelle. Now, remember, it's the mitochondria still. It doesn't have a brain, but natural selection favored it giving up its living identity on its own because it was more likely destroyed, right? Like, it could have come into contact more easily by something else that would have aided, digested it, so forth and so on. But if it became part of a cell, at least the outer part of the cell is still, the membrane or the wall is still protecting it. The other cool thing is that the mitochondria, we know, was an entity in itself because it has its own genetic information. Mitochondrial DNA, which is why mitochondria does not need the nuclear DNA to replicate. And this is why, let's say, if you are working out and your muscle needs more energy, that muscle cell actually can have its own mitochondria replicate itself on the spot. The other really cool thing is mitochondrial DNA is exactly like your mom's. And now, scientists wanted to say, okay, well, if it's exactly, just a little side note, scientists wanted to say, okay, if it's exactly like mom's, that means if some people read their doctrine literally. So what do I mean by that? What I mean by that is there's different doctrines. So we have our Holy Bible. Some choose to read it literally, word by word. And then some choose to acknowledge that, well, the bolded parts or the parts that are in red are the literal direct translations. And then some of the other stories are messages meant to be interpreted contextually. And there's other doctrines, right? I'm referring to our Holy Bible. So you can really kind of take this case elsewhere. But what I'm trying to say, where am I going with this? Where I'm going with this is that if we all have mom's mitochondrial DNA, and the Adam and Eve story was literal, which it's not bolded, which is not in red, so it's not, as it depends on the version of your Bible. It's not in red or bold or whatever. It's not meant to be interpreted literally. And if it was literal, then we would have all the exact mitochondrial DNA because we would have all come from Eve. So scientists wanted to take a look at that, saying that, it's problematic for some who believe that that's a literal story and not meant to be interpreted contextually. And what scientists found was that actually, no, we do not all have the same mitochondrial DNA, but it's interestingly enough, their evidence does support the multi-regional hypothesis, which is mitochondrial DNA is, there isn't a whole bunch of different types, just like, let's say, with nuclear DNA, meaning that there's like handfuls of different mitochondrial DNA. What that implies is that it supports the multi-regional evolutionary hypothesis, meaning that Homo sapiens sapiens were evolving at different parts of the world, some at the same time, some overlapping time, and isolated from one another, which is really cool. And you'll learn more about the multi-regional hypothesis in first-year bio as well. Any questions? Is because it talks about endosymbiosis, the evolutionary hypothesis, a theory. So it's not just a hypothesis, it's a theory. Affirmative. Let me look that one up. And so this is why, I say that regardless of the doctrine that you may ascribe to, it could be problematic if you're told to literally read it word by word and trust by word that it is exactly that case. That's where I'm saying that it can be problematic. And that ends our lesson today. Eukaryotes versus prokaryotes. My other period class, my morning class was super fun this morning because I had a lot more time to go through your mark books and your marks yesterday because we had a longer period. But also what was brought to my attention is all of these personal comments that I'm writing to you. Apparently, you just see your mark. You're not actually seeing the personal comments that I'm writing to you. Is that the same experience that you guys are having? Ever since the first test, you've never seen any of the remarks that I've been writing down. That is just... So, uber, I'm putting it very politely, uber frustrating for me, because that's really where a lot of my time is spent, is writing these personal comments. And so that was brought to my attention in the morning class. So I have fired an email to ICT regarding D2L saying, hello, I'm writing all of these comments, but my students aren't seeing them. I see them. I see that I've published them. So I publish it. Your marks are published, but for some reason, the comments where you lost, let's say, marks, sure maybe they are. That those aren't being published to you. So I'm working on that because I would like to have that at least taken care of by your next unit test, right? Because it does take time. I just think you're like, oh, it's C instead of a G, or somebody else beside you put an A instead of a T, which means the specific change in base sequence that you choose will be different from everyone else. Therefore, the answer key will be varied. That's why there is no answer key for the remaining pages. The first part that kind of walks you through what you're supposed to do, that I just posted that answer key. So that's why I think starting that worksheet